Hello dear friends, welcome back again to another video from my YouTube channel. Dear friends, in our previous video we discussed about the entire process of spermatogenesis. That means we have seen the process of sperm formation inside the testis in the last video. In this video, we are going to understand about the very important concept, that is structure of human sperm. Ultra structure of human sperm. Detailed structure of human sperm. Because this question was repeatedly asked in the competitive exams like AIPMT, NEET, CET, JEE. And the same question was also being asked in the previous year question papers of Karnataka PU board examination for 5 marks. With the help of a neat label diagram, you need to explain the entire structure of a human sperm. Let us start the video. Let us try to understand the structure of human sperm. What is sperm, my dear friends? Let us talk about the basic things of sperm. As we know, sperms are haploid. Why they are haploid? Because they are formed by a meiotic division. Sperms are haploid, flagellated, that means they have the flagella, a long tail. They are haploid, flagellated, motile, male gametes. Male gamete. This is the technical definition of the sperm, my dear friends. Now, these sperms, they were first discovered by a scientist. They were discovered by a scientist. His name is Anton van Leeuwenhoek. Anton van Leeuwenhoek. He is a scientist who observed the human sperms for the first time in the year 1678. In the year 1678. This Anton van Leeuwenhoek, what he believed is, he believed the sperms are the parasites in the male body. He believed that the sperms are the parasites. He believed sperms are the parasites in males. Later, one more scientist came. His name is Spallanzani. Spallanzani in the year 1700. He carried out various experiments to prove that sperms are not the parasite as the Anton van Leeuwenhoek believed. The sperms are the male gametes. He proved the sperms are male gametes. Here you need to remember the names of two very important scientists who discovered the sperms. Anton van Leeuwenhoek in the year 1678, and the same person, Anton van Leeuwenhoek, he coined the term spermatozoa. Zoa means animals. Sperm like animals. This spermatozoa word was first time coined by the Antony van Leeuwenhoek in the year 1678, and the same person is the responsible for the discovery of the sperm, my dear friends. Now, after understanding the historical background of the sperm, let us talk about the size of the sperm. Size. What is the size of the sperm? Human sperm is approximately 52 to 62 microns in its length. This is about the basic things of the sperm. And the sperms can be divided. The human sperms, they consist of 4 very important parts, my dear friends. Human sperms, they consist of 4 very important parts. First one is head. Second one is neck. This neck is very small, negligible, but it is present in the sperm. And third one is middle piece. And the fourth one is tail. They are the 4 very important parts of the human sperm, my dear friends. Let us try to draw the ultrastructure of a sperm and try to understand the various parts and their respective functions. As we have discussed earlier, human sperm consists of a conical shape. It consists of a conical shape nucleus. It consists of a conical shape nucleus. This is the nucleus. This is the nucleus. And the nucleus of human sperm, it consists of DNA, genetic material, and basic proteins. Basic proteins. They are the 2 very important things that are present inside the nucleus of a human sperm. At the anterior lobe of a nucleus, this is the anterior lobe and this is the posterior lobe. Anterior lobe is pointed, whereas posterior lobe has got a flat structure and it has got a small depression here. It has got a small depression. At the anterior lobe of the nucleus, here we'll have a structure like this. This structure, it is actually the acrosome. If you recall my previous video, acrosomes are derived from the Golgi bodies. Acrosomes are derived from the Golgi bodies. Let us label this. They are the acrosomes. Acrosomes. They are the derivatives of the Golgi body during spermiogenesis process, my dear friends. Acrosome, it consists of 3 very important enzymes in it. 3 very important enzymes. First one is hyaluronidase. Second one is neuraminidase. Neuraminidase. And the third one, this is the second one, and the third one is acrosome. They are the 3 very important enzymes present in the acrosome region. Acrosome is derived from the Golgi. Acrosome is derived from the Golgi complex and it consists of 3 very important enzymes: hyaluronidase, neuraminidase, and acrosome. All these enzymes, they help in breaking the egg. They help in dissolving the egg during fertilization. During fertilization, what actually happens is, if this is the egg, if this is supposed to be the egg, here there is a sperm. Sperm want to enter into the egg. If the sperm penetrates into the egg, if it want to penetrates into the egg, the sperm has to break down the egg layer. Without breaking the egg layer, how the sperm made an entry into the egg? It is not possible. So, to get enter inside the egg, the sperm need to dissolve the outer layer of that egg. That outer layer get dissolved by the action of these 3 enzymes. That means these enzymes help in entry of sperm in ovum during fertilization. That is the main function of the acrosome. Acrosome, it is covered with one more protective layer. Acrosome is covered with one more protective layer. This protective layer covers the entire acrosome as well as it covers half of the nucleus. Half of the nucleus, and the entire acrosome get covered by another protective layer. This protective layer, it is called as, I am labeling here, it is called as galea. It is called as galea. It is called as galea. Galea is covering the entire acrosome, as well as it covers half of the nucleus. And the whole thing, it is covered by one more layer, that is nothing but the plasma membrane. That is nothing but the plasma membrane. Plasma membrane. This entire portion, this much part, it is called as the head. It is the head, the first part of the sperm. Head consists of various important structures in it: nucleus, acrosome, galea, and the plasma membrane. And this plasma membrane, it gets a small depression here that forms the neck region. Only this much portion, it is called as neck. It is the neck region. As I told earlier, neck is very small and it is not that much distinct. It is negligible, though it is present. That is why I am mentioning here, it is the neck region. Here, in the neck region, we have got two very important structures. At the base of this nucleus, we have a centriole here. We have a centriole here. Whereas in the neck region, here also we have another centriole. There are two centrioles present in the human sperm. One is at the base of the nucleus and the other one is at the neck region. The centriole, which is present here at the base of the nucleus, it is called as proximal centriole. Proximal centriole. This proximal centriole, it helps in spindle formation during the zygotic division. It forms the spindle. It forms spindle fibers. Spindle fibers during zygotic divisions. After fertilization, zygote is formed and that zygote undergoes repeated mitotic divisions. They are called as cleavages. During cleavages, the proximal centriole, it forms the spindle fiber. That is why it is present at the base of the nucleus, my dear friends. Whereas one more nucleus is present, one more centriole is present. This centriole is present at the neck region. It is present at the neck region. This centriole, it is called as distal centriole. Distal centriole. How many centrioles are present in the human sperm? There are two centrioles are present in the human sperm. One is at the base of the nucleus and other one is at the neck region. The centriole which is present at the base of the nucleus, it is called as proximal centriole, that is helping in the spindle formation during the zygotic division. Whereas the nucleus whereas the centriole which is present at the neck region, it is called as distal centriole. This distal centriole, it forms various filaments like this. It forms the various filaments like this. All these filaments, they are called as axial filaments. They are called as axial filaments. These axial filaments, they form the cytoskeleton. They form cytoskeleton. That means they are acting as a skeleton to the sperm. Cyto means cell, skeleton means the supporting structure. They are the supporting structures formed by the distal centriole. Now, can you tell me the function of the distal centriole. What is the function of a distal centriole? Distal centriole produces axial filaments. Distal centriole produces the axial filaments, whereas proximal centriole forms the spindles fibers during the zygotic divisions. Then these axial filaments, they are of 9+2 arrangement. 9+2 arrangement. It is very, very important for competitive exams. 9+2 arrangement is there in the axial filaments. Now, this entire structure, it is covered with a plasma membrane, and this plasma membrane, it forms a broader structure. This broader structure is the third part of the human sperm, that is middle piece. Middle piece. This is the middle piece of the sperm. In this middle piece, there are various mitochondria are arranged spirally. Mitochondria here, they are arranged spirally. This is all mitochondria. These mitochondria are present in the middle piece region, and they are all arranged spirally. Let us label them. What are they? They are the mitochondria. Mitochondria. We know mitochondria are called as powerhouses of the cell. So these mitochondria are arranged spirally, and this spiral arrangement of mitochondria in the middle piece region of a human sperm, it is called as Nebenkern. Nebenkern. What is Nebenkern? Nebenkern is a spiral arrangement of mitochondria in the middle piece of a human sperm, and these mitochondria, they are approximately 50 to 75 such mitochondria are arranged in the middle piece region, and the main function of these mitochondria are energy source. They provide energy to the sperm, my dear friends. And this entire structure, it gets little depressed here, and it forms a long tail. It forms a long tail here. It forms a long tail. This is the tail region, fourth part, tail of the sperm. And the tail's main function is to help in motility. To help in motility of the sperm. So here the tail is moving, here the tail is showing a movement so that the sperm can easily swim inside the vagina. To show the movement in the tail, we need to have some amount of energy supply. That energy is supplied by the mitochondria. This is the entire structure of the human sperm, and the diagram, the labelings, they are very, very important for competitive exams. The most complicated structure is the head region, because here we have some important parts. Whereas one more very important thing is the tail. It consists of some amount of cytoplasma. Cytoplasma is present only in the tail region in case of human sperm, whereas the remaining parts, they don't have any cytoplasma, and this outer membrane, it is called as plasma membrane. It is the plasma membrane. They are the very, very important labelings, and the diagram is very, very important for board examination, because it has been asked in the year 2014 and 2017 for 5 marks. I hope you people have got an idea about the entire concept of a human sperm. In our next video, we're going to understand about one more very important process, that is oogenesis, the process of formation of ovum inside the ovary. Thank you so much.